Welcome to Study Loop.
On which level is eukaryotic gene expression controlled? By gene expression, we mean the observed protein activity for which that gene uh, encodes for. So here I have just a very simple overall overview of the process. You have DNA that is transcribed into RNA, and you have RNA that is translated into protein. And if anything happens during that entire chain of events that affects protein activity, then you can say that that's a place where gene expression is actually being controlled. So let's look a little closer at each one of these steps. So what I've drawn here in red is DNA. And in the nucleus, DNA wraps around histones. And that's what I've drawn here as these black spheres. The black spheres are actually an octamer of histones, and the histones have these tails that stick out of them. And to those tails of histones, you can stick acetyl groups. And to the DNA, you can add methyl groups. Now, when you do that, that results in a binding of DNA around the histones that is more loose. That means that there is an area, there is a stretch of DNA that is now available to be transcribed that was not available before. And at this level, we have epigenetic control. Epigenetic, epi means above, genetic gene, so you have control above the gene. Okay, so one level of control is the epigenetic control. Now, what I've drawn here on the top with the tata and the gene, it's at the stretch of DNA. The gene is drawn in black, and right ahead of the gene, you have a stretch of DNA that's called the tata box, and it's called that because it has repeat sequences of TA, TA, TA nucleotides. And this is how the polymerase and the entire complex of proteins that helps transcribe the gene, this is how they know where to bind to start transcribing. So you go from the top to the bottom. So right here, transcription has occurred, and the RNA polymerase has moved to the side in the green, and the RNA, which is drawn in blue, has been made. So this is control at the transcriptional level. So you can control gene expression at the transcription level by affecting the affinity of the polymerase and all of these other binding proteins to the promoters or the tata boxes of the genes. Let's move on to the next one. So right here, I have the RNA. Now the RNA, and what's happening here is translation. Okay, because down here we have protein. So we have RNA that is being translated into protein. So this is a chain of amino acids down here. To RNA, these RNA binding proteins can bind that affect the stability of the RNA, making it more stable or less stable. The more stable the RNA is, the more protein you're going to have. So this is controlled at the translational level. Okay, and furthermore, after all that happens, you can still modify the protein itself, let's say by the addition of phosphate groups. The addition or removal of phosphate groups from a protein is a very common mechanism of turning enzymes on or off, making them active or inactive. And at this point in time, the translation has occurred, so this control is post-translational. So as we saw, gene expression can be affected at the post-translational level. It can be affected at the translational level, at the transcription level, and at the epigenetic level. So the correct answer is D, the one that has all of these included in it.